Today, it's my pleasure to chat with Dr. Ronald Petersen. He's a neurologist at the Mayo Clinic in Rochester, Minnesota, where he's also the director of one of actually quite a few funded NIH Alzheimer's disease research centers. And he may want to talk about that for listeners and viewers to know that there's big efforts under the way to try to understand Alzheimer's disease and develop ways to reduce risk and hopefully eventually have effective treatments. So Ron, I've known you for quite a while now because a lot of my work over the years was on some of the cell and molecular biology issues relevant to age-related neurodegenerative disorders like Alzheimer's disease. And we're both Minnesotans. I was actually born and raised in Rochester. I went to Mayo Clinic. There were a lot of doctors' kids at Mayo Clinic, although my father wasn't a doctor. You knew my father. We may talk about that a little later on. And so where did you grow up in Minnesota, and how did you end up? Did you have a parent that was a physician or a scientist?
Mark, first of all, thanks very much for inviting me to have this conversation with you. I look forward to the discussion all day. Actually, I was born in Milwaukee, Wisconsin, but my family moved to Minnesota at a very young age for me, about 6 years old or so. So I really grew up in Minnesota. No medicine in the family. And actually, I just sort of evolved to it accidentally. And as you well know, I spent some time in the biomedical research laboratory of the Army in Edgewood, Maryland, just down the street from you. And that's really where my interest in medicine evolved. I had been sort of a cognitive neuroscientist before that. So briefly, again, grew up in Minnesota, undergraduate school, went to a small liberal arts school in St. Paul called Hamlin University. I'm a Vietnam era person, so that became an element in my overall training and development, such that in my latter days of working, when I was in undergraduate school, I worked at Honeywell in St. Paul, and Honeywell was doing human factors research on man-machine interactions, performance characteristics. And so I was going to go to graduate school in that area, which is now called cognitive neuroscience, but would have been drafted into the Army. So I took a job at Honeywell, working there for a year or so with a critical skill deferment, but that allowed me to enroll at the University of Minnesota in a graduate program. So I did that, and then I actually enrolled in a program in the Army that would allow me to finish my graduate training at the University of Minnesota, but then go into the Army in a laboratory capacity and spend some time. Hence, I finished my PhD at the University of Minnesota, cognitive neuroscience, then went to Edgewood, Maryland for the Army's Biomedical Research Laboratory and did psychopharmacology research there. So the Army was asking several questions about how various drugs affect the mind, in particular in soldiers who are out in the field. So all soldiers who are out in the field, even today, carry with them an atropine auto-injector in case they get exposed to nerve gas, cholinesterase inhibitor kind of gases. Yeah. And consequently, the Army was asking the question, well, if these folks give themselves injections of this atropine, what does it do to their memory, their mind, et cetera? So we started studying a whole host of pharmacologic agents that would affect memory, reversing them, et cetera, et cetera. It became very interesting, such that my colleagues at that time said, if you really like doing this work, you probably need to be a physician. So I applied to medical school, was accepted here at the Mayo Clinic, came here, did medical school, went to Stanford for an internship, back to Mayo for a residency in neurology, and then to Harvard for a 2-year fellowship in behavioral neurology, came back to Mayo 1986 and have been on the staff ever since. So, and now as a neurologist, I'm studying memory, thinking, and of course, Alzheimer's disease is the focal disease there. And if you look back on my career, they say, boy, that was all neatly planned and the trajectory. It was all serendipity, turning a corner here, there, and everything. So it makes sense now. But in fact, I came to studying Alzheimer's disease from a cognitive neuroscience background, psychopharmacology, and then neurology.
Yeah, and that explains you're one of the most prominent clinicians focusing on Alzheimer's disease in the world. You've testified before Congress. You're on many panels that are dealing with issues. Now, one of the areas you focused on a lot, and we'll talk about this in a little bit, I guess, is mild cognitive impairment, and which in some cases, maybe many cases, can presage the onset of Alzheimer's disease, a progressive dementia. So first, do you want to talk about the problem with cognitive impairment and Alzheimer's disease in the United States?
Sure. As you can imagine, it's a very, very common issue. The prevalence of Alzheimer's disease, dementia, mild cognitive impairment is very high in this country. The Alzheimer's Association estimates almost 6 million people in this country have Alzheimer's disease. The numbers vary and how you define it and all that, but it's a very significant issue. And as we, maybe not you, but certainly as I age, I experience these issues of who's that guy I ran into in the grocery store, hey, chief, buddy, how you doing? You worked with him for 8 years and you can't remember his name now. 2 aisles later in the grocery store, there's Bill. How can I forget Bill? So those kinds of things are very common. Multitasking, speed of processing, all these issues slow with aging. But then there's an element of that function that actually deteriorates more rapidly in some individuals. And that's where we get into the neurodegenerative diseases. And among those, Alzheimer's disease is the most common cause of that. But you mentioned mild cognitive impairment. I think that we've studied that here at Mayo for decades, actually. And we didn't invent the term, we didn't invent the construct, but we put some guardrails around it. We're doing a study that you may be very well familiar with, Mark, here at Mayo, called the Mayo Clinic Study of Aging. So as you well know, Mayo is a big institution in a small town. Yeah. And we and the Olmsted Medical Group, the other multi-specialty group in town, basically provide care for everybody in this community. That affords us the opportunity to really study these people, and they're very generous with your time, like your father was, and your whole family. People are willing to contribute their time and effort to participate in research at Mayo. So we've done this study now called the Mayo Clinic Study of Aging, which goes back decades in its previous form, but in 2004, we started it as a population-based study. And by that we mean that we randomly sample people who live in the community, regardless of what other medical issues they have, psychiatric issues, we take all of them. And that affords us the opportunity to study sort of aging in place. Out of that setting, then, many years ago, we observed a group of people who seemed to be forgetful. Now, more forgetful than they used to be, and maybe more forgetful than they ought to be. And that's the key. Again, we all get a little forgetful as we age, but now are you slipping even more so that you're more forgetful than an average 70, 75, 80-year-old person? We observed these people and we kind of classified them separately. And then ultimately, after following them for years and years, we put some criteria around them and called them mild cognitive impairment. And as you indicate, it's often on the continuum of normal aging, memory changes with aging, mild cognitive impairment, and then ultimately dementia. Dementia meaning I'm not thinking as well as I used to, and it's affecting my daily function. Mild cognitive impairment is I'm not remembering as well as I used to, but otherwise I'm okay. I'm doing everything. I'm driving, I'm paying my bills, doing my taxes. To the casual observer, I look fine, but I know, and those around me know, things aren't quite the same as they used to be.
And how do you go about evaluating the cognitive function in a person and rating an individual, based on kind of a normative range of
Yep.
performance on cognitive testing.
So again, because we've been studying this community for decades and decades, some of our former colleagues like the late Dr. Bob Ivnik, Glenn Smith, and other individuals here in neuropsychology actually did normative work in the community. So we can say with reasonable confidence that this 72-year-old woman with 13 years of education should perform thus and so on these various tests. And that gives us a benchmark. It doesn't say yes, no, normal, abnormal, but it sure gives us a good metric now to gauge whether or not this individual is functioning as you would expect he or she to do.
Now, besides testing their learning and memory ability and different cognitive domains, what other kinds of testing do you do? Do you routinely do brain imaging?
Yes. So in the Mayo Clinic study of aging, they're seen by 3 individuals in the study. So a nurse study coordinator goes over the person, interviews them, goes over their medical history, looks at their family history, their medications, and asks permission to speak to somebody who knows them well. So we get another perspective on how that person's doing. That's one input. Second input is from a physician. Physician will do a medical history, look over the medical chart. We'll do a mental status exam, a neurologic exam, and then the physician comes to a conclusion. And then thirdly, we do the cognitive testing. We do 9 tests in 4 cognitive domains. And then we all sit together on every Tuesday afternoon and argue about the profile of this person, whether normal or not. But we also get a lot of personal information. We have the medical histories on all of these individuals because they're our patients. And we also gather fluid biomarkers. So we get blood for DNA, we get plasma, we get serum. We do a lot of imaging studies to get biomarkers for the Alzheimer's disease spectrum, MRI scans on many people, FDG or glucose PET scans that tell us what part of the brain is doing what. And then we do molecular PET scans to see whether there's amyloid and tau present in the brain. Some people even volunteer for lumbar punctures, and we do spinal taps, and we get CSF, cerebrospinal fluid, on these individuals. And again, these people are committed to us. Thank you very much because of their dedication. We follow them along, and often we'll get autopsies on these individuals at the end of the day. So we put this whole package together of clinical evaluations over the years, the biomarkers over the years, the imaging studies, and then we correlate that with the brain autopsy.
And with the 3 imaging, so the structural MRI, you can actually measure the size of different brain structures. And one brain structure that's focused on a lot is the hippocampus.
Right.
And it's a brain region that's critical for short-term memory, mainly because information coming from all of our senses kind of funnels into the hippocampus, and that's where the initial encoding of memory may occur. And then there's some evidence maybe memories are stored, can kind of be transferred.
Networks, yeah.
Network. And then the other is the functional MRI, right, where you're looking at actual activity, neural network activity.
Right. Right.
And then the PET imaging is interesting. And you mentioned this radio-labeled form of glucose. Now, it's been known for a long time that brain cells seem to have a problem in using glucose in MCI and in much more so in Alzheimer's. And I had done an earlier podcast, Steve Cunane, who you may have come across.
Sure.
And he did PET imaging combining, looking at glucose utilization and ketone utilization. The ketones are derived from fats. Right. And he has some very nice data showing that when he switches people to a ketogenic diet or gives them a kind of a fatty acid precursor to the ketones, the brain, this in normal people, the brain cells switch. They switch from using glucose to ketones.
Mhm.
And he has some evidence now that people with MCI, the brain cells can still use the ketones apparently at normal level, even though they're having problems using glucose. So this is, when we get to talking about therapies, we might circle back to that.
Sure.
It's one approach. The point is there's an energetic problem.
Yeah.
in the brain.
Yeah. What we look for in FDG PET scans is the pattern of glucose hypometabolism. And a typical Alzheimer's pattern, again, it's not definitive, not diagnostic, but typical, is a lateral parieto-temporal hypometabolic pattern, also posterior cingulate, precuneus. They can be pretty specific that this is an Alzheimer's process going on. There are other patterns, of course, that will tip us away from Alzheimer's disease, make us think of other disorders. If there's a lot of posterior hypometabolism, parieto-occipital, we may think about a Lewy body component. Visuospatial impairment is very prominent early in Lewy body disorders, not so much in Alzheimer's disease. And of course, frontal hypometabolism is very common. Frontotemporal dementias, various forms of primary progressive aphasia have different metabolic signals. So it's very useful tool in terms of telling us what might be going on, and in particular, whether multiple things might be going on. And some of our colleagues here at Mayo have, looking at literally thousands of these PET scans over the years, have developed a model they call the state viewer, which really puts a person's metabolic pattern in a broad scheme of other individuals who have known various clinical entities, maybe normal aging, Alzheimer's disease, Lewy body, frontotemporal, and puts this particular metabolic signature, if you will, in that total scheme to let the clinician know, gee, this looks like maybe Alzheimer's, but it's kind of close to Lewy bodies as well. So it may in fact represent some of both. So I think we're becoming increasingly sophisticated in our use of these functional imaging modalities.
Now, when my father, we first, so the way I first noticed he was having problems was, and I think this is typical, of course, I live in Maryland, he's back in Minnesota. And when I talked to him on the phone, just even in a short conversation of say 5-10 minutes, he would ask me the same question more than once. You know, sometimes three times. Yeah. And of course, my sister is closer to, physically close up there in Rochester, and she was, but you know, it was clear his short-term memory was deteriorating, but he was living on the farm and he seemed to be initially okay. You know, he was managing the things he needed to do, taking care of himself, driving. And then when we brought him to you and you did the cognitive testing and so on, you diagnosed him with probable Alzheimer's disease. Right. Why probable? Yep.
A very good question, Mark, and it really gets into how the field is evolving in terms of diagnostic capabilities. So again, with your permission, when I initially saw your dad, you described it adequately. He had a difficulty with new learning and retaining recently experienced information. So again, the hippocampus was absolutely crucial in laying down new memories and recalling recently experienced events. And he was having trouble with that. And his hippocampus was atrophic, certainly smaller than we would expect even for age. Again, having done these on thousands of people in this community, we have a pretty good index of what is a normal-ish looking hippocampus for an individual of a certain sex and age. And your dad's was small. So, but we were at that time living with the criteria that had been developed back in 1984, actually by Guy McCann at Hopkins and colleagues in the field, as to describe the diagnostic criteria for Alzheimer's disease were what I was describing earlier of gradual impairment in memory, other cognitive functions being impaired. Then it eventually affects your daily function. We call that dementia. And we'd say, okay, then we do a bunch of what was called rule out exercises. So we looked at your thyroid, your B12 level, as scanned to make sure you didn't have hydrocephalus or a stroke in your thalamus or a brain tumor, something else going on. And if we didn't find that, we'd say it's probable Alzheimer's disease. We couldn't say definite Alzheimer's disease until the person went on, passed away, we did an autopsy and saw plaques and tangles in the brain. Then it's definite Alzheimer's disease. But this was 1984. Served the field well for decades, actually. But in about 2011 or so, the NIA and the Alzheimer's Association convened some committees to say, gee, haven't we learned more about the biology of this disease? Haven't we really discovered that these biomarkers now can give us some information about the underlying etiology of this clinical syndrome, say MCI? And so the committees developed some criteria, 3 sets, one for preclinical Alzheimer's disease, meaning people are clinically normal, but may have positive biomarkers. MCI, have the MCI syndrome, plus minus biomarkers, and then the dementia stage, again, dementia clinical syndrome, plus or minus the biomarkers, and we come up with low likelihood, intermediate, high likelihood, and things of that nature. So that moved the field forward. Then in 2018, NIA Alzheimer's Association again convened a committee. Shouldn't we update this now, 7 years later? And our colleague here at Mayo, Cliff Jack, led the committee, and they published a very important paper in 2018, still percolating in the field. And it was research recommendations defining Alzheimer's disease now as a biological entity, meaning you can't have Alzheimer's disease if you don't have amyloid and tau in the brain, either by biomarkers, fluid, imaging, or at the time of autopsy. But importantly, it was irrespective of the clinical state of the person. So now we're talking about people who are clinically normal, who could be labeled as having Alzheimer's disease if they have biomarkers positive for amyloid and tau. The thinking was, let's put Alzheimer's disease on the same playing field as other medical disorders. In particular, if you've got a cancer, if you've got a positive prostate biopsy, you have prostate cancer. You have a positive breast cancer, a breast biopsy, you have breast cancer. Irrespective of your clinical state, you have that disease. So the thinking being, if we define Alzheimer's disease in that fashion, it'll help the development of clinical trials, treatments, and hopefully moving to the preclinical stages to prevent the disease. So that was the thinking. So we've done away with the probable, possible, definite Alzheimer's disease. If in fact these criteria are adopted by the community, then the disease will be based on its biologic characteristics.
And with this new biological characteristics approach, does that imply that someone who meets criteria for that, based on the biomarkers and imaging, would likely develop severe cognitive impairment if they were to live long enough?
Right. So that's an important point, because detractors of this position will say, I'm not so sure that that's the way the disease should be defined, because people can live through their life or later life with a head full of amyloid, die clinically normal, and never become demented.
But then going back to your cancer parallel, my father actually had prostate cancer. He didn't die from prostate cancer.
Right. He
right.
died with it.
So
yeah.
That's, it's similar. You can have prostate cancer and not die from it.
Exactly.
Yeah.
But the people who are not fond of the amyloid cascade hypothesis would say that amyloid is sort of a bystander. It comes along, but it's not etiologically involved in the sequence of events. Others would contend, no, amyloid is the earliest manifestation biologically of the disease. Subsequent downstream effects on tau and other proteins then lead to the degeneration and ultimately clinical impairment. And again, my colleague Cliff Jack here developed a set of curves that have been popularly used in the field over the past 12 years now, that lay out this cascade of events. And by and large, it's been substantiated by the data in the field that this is the sequence of events that occurs. And the importance for treatment implications, of course, is that presumably this amyloid deposition, amyloid misprocessing, if you will, occurs many, many years, maybe a decade or two prior to clinical symptoms. Hence, that gives us a great window for intervention to intervene, reduce amyloid misprocessing one way or another, not easy, but treat it and prevent the clinical symptoms from manifesting themselves. But that's still up in the air right now in the field, and we certainly haven't demonstrated. But that's kind of the thinking about it in terms of this cascade of events.
Okay, and then as you mentioned, my father was enrolled in your research study program, and when he passed away, Dennis Dixon, a colleague of yours, and his people took pieces of my dad's brain and looked at them under the microscope. And his case, my understanding, was a little atypical in terms of the pathology.
It was atypical in a sense at that time, the way we were viewing things. What it really meant is Petersen was wrong, actually, all along. So what your dad had was what we're seeing very commonly now in recognizing as the multifactorial nature of contributions to cognitive impairment. So Alzheimer's disease is very common, even if you define it biologically. It's very common in the brain in older people, but it doesn't exist alone. There's often other neuropathologies present. Alpha-synuclein that produces Lewy bodies, things we see in Parkinsonism, dementia with Lewy bodies. We see TDP-43, another protein that is commonly deposited in the medial temporal lobe, the hippocampus, and can cause something called hippocampal sclerosis, which is severe impairment of the hippocampus, can give prominent memory disorder that really looks like incipient Alzheimer's disease, but is largely due to the deposition of that protein in the hippocampus. And then vascular disease, microvascular disease, not big time strokes necessarily, but microvascular disease. Almost everybody will have that if they live long enough. All of us will have tau deposition, probably in the medial temporal lobe, if we live long enough. So your dad was really presaging what's happened, what's come to be recognized in the field, that these disorders really are multifactorial. And you put in the TDP-43, a little bit of diffuse amyloid. It looks like Alzheimer's disease clinically, but in fact, it may be due to these multiple other co-pathologies that are going on, such that we now say that is the rule. Multiple co-pathologies, the rule, not the exception.
On the biomarker front, so you mentioned you can look at this abnormal form of tau in the cerebrospinal fluid and even blood, and there's a certain form of amyloid or a certain number of amino acid form, that so-called A beta 42, seems to be elevated. And a colleague of mine who was actually in my laboratory when I was lab chief at National Institute on Aging, Dimitros Kapagiannis, has been developing what are called extracellular vesicles, or exosome-based approach. And so all cells, including neurons in your brain, they secrete these very tiny nanoparticle vesicles. And some of these seem to be getting into the blood, and by using antibodies that are specific for neurons, Dimitros is able to pull out of the blood, the circulating pool of these vesicles in the blood, those that he thinks are coming from neurons. And he's been looking at some of the same biomarkers and others. So I think that's the cancer field developed that, and they called it liquid biopsy.
Yeah,
so
interesting.
The idea was, in that case, these vesicles were being secreted by the cancer cells, and they had particular molecular markers on them, and they could look at them. And in the case of brain disorders, the idea is that there's abnormalities in these vesicles released into the blood that can be looked at.
Right,
So,
right. Yeah,
anyway.
the field of fluid biomarkers is really taking off, in particular with regard to plasma biomarkers. So as you mentioned, I discussed earlier, one of the biomarkers you can use that's pretty definitive is CSF, cerebrospinal fluid. And you look for low levels of A beta 42 or a low 42 to 40 ratio, or a ratio of phospho-tau, p-tau to 42. All of these are very good at detecting amyloid and tau in the brain. But where the field has moved in the last 5 years or so is the availability of detecting many of these same features in the blood. I think with increased mass spectrometry and immunoassays, you can now detect for p-tau 181, p-tau 217, 231, 205, a variety of these different species now in the blood that are really indicative of things going on in the brain. And we and a bunch of other people have done studies showing that these blood levels of, say, p-tau 181, 217 correlate very well with PET imaging for amyloid and tau. So the field now is poised to see, one, do these blood markers actually predict whether you're going to be amyloid positive on a scan, because that has implications for your enrollment in a treatment trial. Because right now, if you have to do PET scans or spinal taps on everybody to get into a treatment trial, it's expensive, it's inconvenient, etc. Blood will maybe give you the window of opportunity to really take people who are highly likely to be positive on these. So that's one use that's really come to the fore for the use of plasma biomarkers. The second is, can we actually make the diagnosis? So if we buy the biologic diagnosis I discussed earlier for Alzheimer's disease, can we do that with a blood test? Can we say, yes, no, you have Alzheimer's disease or not? I don't think we're there yet with that kind of specificity, but it certainly is on the horizon. And then thirdly, using the blood biomarkers to track potential treatments. So you do a plasma biomarker before you start the therapy, you give the therapy, a year later, 2 years later, you do the blood test again. Has the blood biomarker moved with regard to the treatment as you would expect it ought to? So maybe we can monitor therapies with blood biomarkers. So all of these areas are being investigated right now, and I think hold excellent promise.
Let's move on to risk factors. And I guess to start with, Ron, why don't you talk about inherited Alzheimer's disease? Because whereas most cases, such as my father, for example, there's no family history or no known genetic cause, but there are a small number of people who inherit genes that can actually cause a disease.
Right. So generally, we speak about Alzheimer's disease from a genetic perspective in two forms: either autosomally dominantly inherited, that rare form where it runs in families, or sporadic Alzheimer's disease. So in the familial Alzheimer's disease situation, there are these autosomal dominant gene mutations, presenilin-1, presenilin-2, APP, that in fact, if those mutations are present, you will get the disease, meaning that the penetrance is 100%. And you often will get it earlier in life. So people in these families often get the disease in their 40s and their 50s. And you look back through the family tree, and 50% of the people in the generations have the disease manifesting early in life. Fortunately, as important as these are for studying the biology of the disease, potential treatment interventions, it really only pertains to maybe 1-ish% of people with Alzheimer's disease. So the vast majority is the so-called sporadic form. And here now we look for what are called susceptibility polymorphisms. So various gene abnormalities that will increase your risk. Doesn't mean you're going to get it. It's not autosomal dominantly inherited, but it does increase your risk. And the major player here is apolipoprotein E. And ApoE comes in three isoforms: E2, E3, E4. You inherit one form from mom, one from dad, then you get the combinations of 2, 3, 3, 3, 4, 4, 3, 4, et cetera. And that determines to a certain extent your risk. In particular, if you inherit the E4 variety, your risk is increased. So if you have one copy of 4, your risk is increased maybe 3 or 4-fold over the general population. If you're unfortunate enough to get a 4 from mom and a 4 from dad and you're 4-4, now your risk is up maybe 10 to 14 times over the population, and you may get it earlier in life as well. Not 40s and 50s, but it does move earlier in the curve, the age curve. So that's the most prominent. But there are dozens and dozens of other susceptibility polymorphisms that are very infrequent in the population, but do in fact have an impact on your risk of getting the disease. But again, they're relatively rare in the population. But what people are doing now is putting together what's called a polygenic risk score. So they're taking these various dozens and dozens of mutations, putting them together in a combinatorial fashion to actually predict your individual likelihood based on your genome of getting the disease. Now, the biggest player in that whole set, of course, is still apolipoprotein E, but that plus these others may, in fact, alter your risk of getting the disease. So, in general, Alzheimer's disease does run in families, like most other diseases, heart disease, cancer, diabetes, they run in families. So if you have a first-degree relative, mother, father, excuse me, mother, father, brother, sister, who has, again, preferably, let me get a drink here. Excuse me, preferably autopsy-proven Alzheimer's disease, then your risk is up again, something like the ApoE 3 to 4 times over the general population. But again, most likely you're not going to get it, but your risk is increased, just like with other major medical issues.
Yeah. And I don't know if we should talk about genetic screening or not, but what about diet and lifestyle factors? So you talked about genetic risk factors. Are there any diet and lifestyle factors that seem to affect one's risk?
I think, to be honest, I think lifestyle factors can influence perhaps rates of development of, I'll say, cognitive impairment. Less certain about Alzheimer's disease specifically if we define it biologically. But clearly cognitive impairment, the development of dementia, can be influenced by lifestyle factors. I think the strongest data exist in the area of aerobic exercise. So I think we make recommendations of aerobic exercise, ballpark figures, 150 minutes a week, 30 minutes 5 times, 50 minutes 3 times. And this could be brisk walking, could be jogging, swimming, something of that nature. But getting out there and doing that on a regular basis probably does alter the trajectory of cognitive problems developing, if they're going to develop in life. We also talk about intellectual activities, keeping your mind active, doing things. I'm not sure I can endorse any particular brain games or anything of the like, but I think those kinds of activities are probably good for the brain. Again, I usually suggest to patients that they engage in some activities that they enjoy. So it's kind of fun doing this. And some people love Sudoku and other crossword puzzles in the New York Times and whatever. And I think those are just fine, especially if you enjoy them. Thirdly, when we talk about diet, I think the safest thing here to say is perhaps what's good for the heart is good for the brain. So a Mediterranean diet is probably a good example. And there are variations on that, the MIND diet and a variety of others that generally are probably beneficial for cerebral function as well as cardiac function. And then finally, we talk about staying involved in your networks, your social networks, communicating with people, avoid the tendency to withdraw, and get out there and be social with your family and other. I think those kinds of activities are generally well recommended. One important area that has really come to the fore in the past 5 to 10 years, maybe, is sleep health, sleep hygiene, because sleep becomes disrupted in aging and clearly in neurodegenerative diseases. So being mindful of sleep hygiene, avoiding stimulants and things that in the evening that are going to keep you awake. Watch for sleep apnea, very common in aging. Lax tissue in the oropharynx can collapse, causing apneic spells. That is a treatable entity, clearly impacts not only your cardiovascular health, but your cognitive function during the day.
Yeah.
So you can, if you're, if you like take naps all the time, naps are not necessarily bad, but if you're doing too many of those, it's probably because you have disordered sleep at night. So I think sleep hygiene is another very important aspect. So we talk about these lifestyle recommendations constantly. I think they're important. Are they going to prevent anything? I'm less convinced of that. Are they going to alter the amyloid and tau proteins? I don't know about that, but I think they may, in fact, affect your cognitive function and flatten out some of the trajectories over time.
Okay. What about for someone who has mild cognitive impairment or maybe early Alzheimer's disease now in terms of treatment?
Yes. So, over the past year, of course, we've all been embroiled in this controversy regarding the accelerated approval by the FDA of the drug aducanumab for Biogen in June of last year. And it received accelerated approval. And by that, the FDA meant that they thought there was convincing evidence that the drug reduced the amyloid plaque level in the brain, and that reduction in amyloid plaque level was, quote, reasonably likely, unquote, to have an impact on cognitive function in the future. But they also indicated that the clinical data that were presented in support of this drug were not sufficiently convincing at this point in time to give the drug full approval. So they gave it accelerated approval and charged the sponsor, the company, with the obligation of doing an additional study to demonstrate that, in fact, reducing amyloid levels has a clinical impact down the road. So, if you want me to go into details about the controversy, we don't have to do that. But part of it was the fact that the two studies were stopped prematurely for a futility analysis, indicating that approximately 50% of the way into the study, they had predetermined that they were going to have an independent committee look at the data, unblind themselves, look at the actual data, and make a determination as to whether this study, if it went on to completion, would have a reasonable chance of being positive. And the committee concluded no. They said, I don't think this study is going to be positive. Therefore, we recommend stopping it now. Well, again, like in too many details, they did that. They stopped the study, but additional data were acquired during that decision-making period, additional 3 months of data. And when the company looked at the additional 3 months beyond the data that the futility committee had evaluated, they saw a signal. And in fact, they concluded that one of the studies was trending positive, one of the studies was trending negative, went to the FDA, said, would you entertain an application from us? The FDA said, yes, show us the data, and hence everything else ensued. And people said, you never should have gone down that road, never should have given it approval, the clinical meaningfulness was not there, blah, 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 all that. Nevertheless.
And was it considered in light of there were several previous trials of amyloid antibodies or immunization in Alzheimer's, and which my understanding is they saw, at least some of them saw some shifts in the biomarkers, but did not see any clear effect on cognition.
Yes and no. Yes,
Okay.
there were numerous studies that previously had been done on various aspects of amyloid, some secretase inhibitors, beta secretase inhibitors, gamma secretase inhibitors, other forms of antibodies that basically moved some of the fluid biomarkers. So the beta secretase inhibitors did in fact alter some of the fluid biomarkers, but none of them removed plaque from the brain. Okay, so people would say, yes, those studies were negative, but they really didn't do what they were biologically intending to do, is one argument. Now, the FDA also looked at the data, in addition to aducanumab, the data that had been published from other antibody studies that in fact showed the ability to lower amyloid in the brain. Lecanemab, donanemab, gantenerumab, all are other antibodies that are being studied right now that do lower the amyloid level in the brain and are showing a subtle, maybe modest clinical impact. And so the FDA said, you take aducanumab in the context of those newer antibodies, and I think there's sufficient evidence now to warrant at least accelerated approval. So that was, the part of the controversy. But as we know, various things have happened, and basically aducanumab the cost is prohibitive, and it's really not being used to any significant extent. But the other three drugs I mentioned are going to have readouts in the latter half of this year or the beginning of next year. So we should get fairly definitive evidence one way or another as to whether lowering amyloid plaque level actually has a clinical benefit.
Now, you do prescribe drugs that some of the earlier drugs that were developed that there was some evidence may have at least some small but significant effect on cognition or possibly even cognitive decline. And, and they focus on really two neurotransmitter systems. One's acetylcholine.
Correct.
And the other is glutamate.
Yep. Yep.
You want to talk about
that? Yes. So there are 4 drugs, other drugs out approved, full approval by the FDA for the symptomatic treatment of Alzheimer's disease. And as you say, Mark, 3 of them are cholinesterase inhibitors. So donepezil, rivastigmine, and galantamine are all cholinesterase inhibitors. And what that does is keep the amount of acetylcholine available at the synapse for a longer period of time. So it blocks the breaking down enzyme, if you will, and keeps the cholinergic system functional for a longer period of time. But it is symptomatic.
That goes way back to when you were at Aberdeen Proving Ground and
absolutely
with a drug called scopolamine.
Scopolamine, right? Exactly. Scopolamine was a kissing cousin of atropine, and we blocked the cholinergic level using atropine and scopolamine, and people became densely amnestic with that. You reverse that with something like physostigmine, and in fact, the memory function comes back. So yes, 40 years ago, we were working on the same kinds of mechanisms. So, but these drugs and the memantine, the 4th one, which, as you say, works on the glutamatergic system, NMDA antagonists, are all meant to keep these neurotransmitters active for a longer period of time to help with the symptoms, but they do not have any impact on the long-term outcome of the disease. They're not what are called disease-modifying drugs, which the monoclonal antibodies are called disease-modifying therapies.
Now, most of the neurons that die in Alzheimer's disease, this is definitely true, use glutamate as their neurotransmitter. Hippocampus, cerebral cortex. Right. And my understanding, there's evidence that, interestingly, there's actually aberrant hyperexcitability of these neural circuits that degenerate. Right. Is it true that people with Alzheimer's have increased incidence of seizures?
There is a slight increase in that, yes. There is a slight increase in the propensity to have seizures if you match them for age-matched individuals. It is true, and it could very well be due to the hyperexcitability. Also, there's a notion of, is there compensatory mechanisms? So when certain systems start to fail, do other systems try to compensate for that? And whether that's another mechanism that may lead to the hyperexcitability. Yeah.
And what do you see down the road in terms of, you talked about the antibodies to amyloid, and very early on in that research, there's this idea that you could actually immunize someone with amyloid. Right. In theory, even before they have any symptoms, and reduce the accumulation of the amyloid and the consequences of that. have there been clinical trials based on immunization?
Yes, so literally 20 years ago, around 1999, 2000, so-called AN 1792 was the first active immunization study designed to do exactly what you outlined, Mark. That is, knock down the amyloid levels before they even rise and cause symptoms. Now, back then, that study had to be stopped early because of the runaway inflammation in some individuals, such that they actually got encephalitis because of the overstimulation of the immunological system. Nevertheless, the construct is still viable. Other active vaccines have been entertained, and the monoclonal antibodies that are being studied now are really in the same ilk, but are now talking about passive antibodies rather than immunizing people to raise their own antibodies. But ultimately, you'd think that that would be a viable path if, in fact, the passive immunization proves effective. And so I think where the field is headed, hopefully, is if these current monoclonal antibodies show some signal of clinical efficacy at the symptomatic stage, the mild cognitive impairment, the mild dementia stage, we see some stabilization of the condition. The natural extension of that would be to go back to where people are amyloid positive, but clinically normal, the so-called preclinical area. And there are studies underway right now, actually, that are looking at that with some of these different antibodies to see if we might have an impact on the development of symptoms, the rate of development of symptoms in those individuals. So that would be the natural extension of these. And then beyond that, active immunization, so that a person doesn't have to come in for an IV infusion every month or whatever time frame is necessary for these drugs. So I think that's the real extension. And then I close with finally saying, as I was mentioning earlier, and your dad being an excellent example of this, that it's really going to be multiple pathologies that contribute to this. And where the field is headed now is to develop biomarkers for all of these different pathologic entities, and hopefully then develop therapies that are specific for each of these path entities. And we will need combination therapy down the road to treat these disorders, not a single drug, a single pill, or anything of the sort, but it's really going to be a combination of therapeutic interventions, much like we do now with, say, hypertension. When somebody has elevated blood pressure, we treat it. How do we treat it? Well, could be a diuretic, could be a beta blocker, could be a calcium channel antagonist, could be an angiotensin receptor blocker, different mechanisms, all getting at that same symptom of elevated blood pressure. If we take cognitive function or dysfunction as that single measure of clinical performance, now we have to come at it with a variety of different probes to see what you have in your brain. I'm blue skying here, but speculating that down the road, you may go in to see your primary physician, and he or she will get your lipid screen to see how your HDL, your LDL, your triglycerides, and your cognitive screen in the blood to see if you have amyloid, if you have tau, alpha-synuclein, TDP, and therefore, here's what we're going to do about it, and we treat with this combination therapy. I don't know if that's going to happen, but I can certainly think that that might be a positive outcome of this line of work.
And I want to end on the diet and lifestyle thing. Now I was thinking about, so clearly with cancers, many cancers, heart disease, diet and lifestyle have a big impact. And those are age-related, largely age-related diseases. But from what you said earlier, you don't think diet and lifestyle has an impact on the brain as it does on these other organ systems. Is that
I will. If I said that, I should restate that by saying
no. That wasn't exactly what you said. But that was kind of
your take home from it, which I probably misled you then. I think it does have an effect on the brain. Whether it affects the underlying biology of Alzheimer's disease, I'm less convinced, but I think it definitely has an effect on the brain. And that's why I'm saying that if people follow these lifestyle recommendations, you may very well affect your cognitive aging trajectory. What's the mechanism for that? probably combination of factors. But no, I definitely recommend lifestyle modifications if at all possible.
Okay, Ron, we covered a lot of ground. I would like to say one last thing about my father. So, as you know, my mom died fairly early when she was 67. And so my father was living alone on the farm.
Mm-hmm.
So his daily, and he was a Olmsted County attorney for years. Actually, he succeeded a very famous Olmsted County attorney named Frank Kellogg, who went on to be a senator and Secretary of State and Nobel Prize winner. But anyway, so at one point, my dad was really intellectually sharp, right?
Sure.
But then when he retired early at 60 years old. And after that, he didn't read much, because all his reading before was, he'd be up late at night going over cases and so on.
Sure.
And then when mom passed away, he was by himself. And I think that, he had like reduced stimulation. And that, I guess, as a guess, may have kind of accelerated the onset of his cognitive problems.
So of the measures we discussed earlier, he probably lacked some intellectual stimulation, partly because he wasn't working, wasn't reading, and he didn't have the social stimulation from your mom to interact. So two of those four pillars may have been missing for him, or at least diminished.
Yeah. So that's a good way to leave people with, this notion that there are some things you can do and to look out for in your relatives and friends, in their lives. Okay, Ron.
All righty, Mark. Thanks so much for inviting me. I enjoyed the conversation as always with you, and I wish you well.
Me and my wife plan on being up in Rochester sometime in August.
Okay, very good. Give me a buzz. I should be around for part of the month anyway. Okay.
Okay, right.
Thanks again. Okay, take care. Bye-bye.